Good afternoon, everybody. We hope you had a chance to enjoy the poster sessions and enjoyed the session with our editors. We'd like to kick off this afternoon session now. I have the privilege of inviting Professor Craig Mello from UMass Medical School up to begin this afternoon session. So if I could invite up Dr. Mello, and he's going to be talking to us about post-transcriptional regulation of mRNAs by Argonaut risk complexes. Thank you.
Hello, everybody. Thank you. I want to thank Angela and Phil for putting this together, and I know how much work it's been. It's really great to see so many friends from all over the world here and get to show you where we work. It's a fun place to be. And we are hiring, so while you're here, I think we're hiring at every level, right, Chancellor? We definitely are looking for talented people, and that includes in a lot of the labs here. So if you're interested, please. And I want to start just with a few thank yous. Of course, thank you to the worm. It's still got a lot to teach us. And basic science, there's still a lot that this little animal has, a lot of secrets it has that I hope we'll learn about in the future. And this is actually an old picture of the lab, but I wanted to get a thank you to the lab up front. They are great. They are so diverse and free-thinking. They're kind of a pain in the neck sometimes. I can't always get them to do what I want them to do, which sometimes is a good thing. I'm going to tell you a little bit about our work on the worm, and but everybody's heard about CRISPR now, and RNA interference, used to be the hot thing. And I recently got this email, in fact, last Friday when I was getting ready to talk, and they read my paper, so I was like, oh wow, they read my paper, that's nice. And then they said, are you still using RNAi for your And I love this. Look at this. Upgrade your experiments with CRISPR. and for only $2,500 now, you can do a cleaner experiment. All right. So, gee. That was tough. But CRISPR is really, really cool, of course, and I love CRISPR. But the worm and the things that it's doing with Argonauts and how Argonauts are functioning, particularly in the germline, it's like science fiction. It is so cool and really surprising, because what the animal is doing with its Argonauts, they're localized in these perinuclear foci that are so beautiful. My mom, who's an artist, took one look at them years and years ago, and she said, Craig, you should work on those. Those are going to be important. Well, guess what? Like 25 years later, that's where all the Argonauts are in the germline. They're in these beautiful perinuclear halos in the germline. And I'm going to just tell you, because I know otherwise I might really just go over, that what the animal is doing with its Argonauts and its small RNA pathways is it's creating memories of gene expression, and it's transmitting those memories in the germline. And you can't make this stuff up. It's like science fiction. And when we publish papers on this, the reviewers often don't believe the papers we've already published that preceded the one that they're reviewing. So it's actually, anyway, we didn't, I want to just take you a little bit through this because it's so bizarre, you really almost can't believe it. So this Argonaut is targeting all of the self-mRNAs. And we believe it's functioning as a memory of self-mRNA expression. This Argonaut, and it's got several paralogs, are targeting genes that are undergoing silencing, like transposons and transgenes, and I'll tell you a little bit more about those. And then this Argonaut is the peewee Argonaut of the animal, and it's functioning as a scanning factor that identifies sequences that haven't been expressed previously in the germline, identifies them as foreign because they're not targeted by this Argonaut, and then initiates the recruitment of an RNA-dependent RNA polymerase that amplifies the silencing signal and loads this Argonaut, creating a memory, a lasting memory of a new foreign entity. Try to do that with CRISPR. CRISPR actually does something very much like that. But in fact, what CRISPR does is it captures a piece of the foreign element and uses that to make the guide RNAs, of course, that are functioning in this silencing pathway. What the worm is doing is remembering everything that it expressed previously, remembering it all, so that it can innately recognize something as foreign without having to be exposed to it. That is really cool. And we very, very reluctantly came to the conclusion that that's what this animal is doing. In fact, though, we started working on this all the way back in 1999. We started working on this problem. And when we tried to publish we published the paper in Nature with Andy saying that double-stranded RNA triggers this gene silencing phenomenon. And yet we had no idea about the mechanism, so immediately we started hunting for the genes involved. So we did a screen and we found hundreds of mutants and started mapping them and trying to clone the genes. And we got into this big rush for one reason or another. Another lab had cloned a gene involved in transposon silencing, and we found out that some of our genes were involved in transposon silencing too, and we told them, and we were communicating, and we had parallel papers in submission, and we hadn't cloned any of the genes, and we proposed this crazily complicated model based on genetics. So the genetics said that RNA interference functions downstream of double-stranded RNA involving these genes, RD1 and 4 and a couple of others, and then it converges on a pathway that is shared by transposon silencing, but has a distinct trigger. And everybody in the field, including the other paper that had been submitted in parallel, was suggesting that what transposons do is make double-stranded RNA, and that is why transposons silencing shares this pathway, because double-stranded RNA is the trigger for transposon silencing as well. The idea being transposons could insert opposite orientations and get transcribed in both orientations, form double-stranded RNA in the cell, and then through the dicer pathway, et cetera, initiate the silencing. So our paper got rejected. The other paper was going to get accepted because it had a very nice linear model. And actually, I want to show you this slide because, I don't know if you guys follow current events, but let me tell you something. Humans are not very smart. We like really simple models, okay? Really simple models. And we're comfortable with simple models. So are, editors and your reviewers, et cetera. So this is the kind of model for RNA interference that, I think most people really like. Some of you have probably seen this before. Here's a double-stranded RNA. And here are the defective genes. This actually aired on CBS at the 6 o'clock news, okay? Again, Cas9 cannot do that, I guarantee you. So we had this really complicated model. Nobody liked it, but we cloned the RD1 gene, and luckily for us, that was enough to get us over the hurdle. And so we got this paper published in Cell in 1999 that basically described this crazy model in which the RD1 gene, which turned out to be an Argonaut, is involved in the double-stranded RNA response, but there's some other pathway, some other mechanism that leads to transposon silencing. We didn't know what the trigger was, but the genetics had already told us that the trigger was not double-stranded RNA. So how is transposon silencing triggered? So here's the canonical pathway for double-stranded RNA. You have dicer, RD1, RD4 functions up here, and a few other things. And then you have these WAGOs down here. You have an RDRP. This is why worms have such super duper RNA interference, because most organisms have this part of the pathway, but the worms actually can use this RDRP to massively amplify the silencing signal. And this can actually explain why RNA interference in the worm is inherited over multiple generations, because these argonauts are in the germline. So we thought transposon silencing involved some independent trigger, but that it clearly shared this machinery, because those mutants, when we found them, had transposon mobilization, whereas these mutants up here didn't mobilize the transposons. So that was the genetic evidence that transposon silencing is distinct from the RNA interference in terms of the trigger. But what is the trigger? Just a little bit of background. One of the things you can do once you know that the cell is using a search engine, you can start to read the search queries that the cell is sending into its little search engine. So these argonauts are searching for stuff. What are they searching for? So we IP'd different argonauts and we cloned and sequenced the small RNAs. And this sort of created this incredible weird mystery. By far the most abundant species of small RNA that you detect associated with argonauts in the worm are small RNAs that are antisense to the expressed mRNAs. Genes that are normally expressed in the germline are targeted by this argonaut by small RNAs that are made by RDRP and loaded onto this argonaut. And again, as I told you, transposons and pseudogenes, et cetera, are being targeted by these. These are old numbers, but basically almost every mRNA is targeted throughout the spliced transcript by these RDRP-derived small RNAs. So the transcripts get templated by RDRP to produce antisense small RNAs, and moreover, these argonauts, Caesar1 and WEGA1 and some of the other WEGAs as well, and their small RNA cofactors are loaded into both the egg and the sperm. There's a lot of these argonauts and their guides in the mature gametes. So the animal is literally passing these signals down to the offspring. So the breakthrough for us and for other people also working on silencing these silencing mechanisms in the worm, some of them are in the audience, was that we had transgenes. So we could explore these. We can't put a new transposon into the worm, but we can put a transgene into the worm. And to the worm, that presumably is like seeing a new transposon for the first time, at least in some respects. So that's what we did. And one of the things that we noticed is sometimes they're off and sometimes they're on. And like everyone else, at first, we were simply throwing away the ones that were off because, when you make a transgene, you want it to be on. And so you just keep trying until it's on, and then you keep it and you work on it. That's what everybody does. And weirdly, we're in a silencing lab, and it took us, a couple of years maybe before we realized, hey, these might be silenced instead of just not on. They might actually be actively being turned off. And Masaki got this idea, and along with me too, went ahead and did this very simple experiment shown on the next slide. Actually, one more additional point that I think is interesting. There's a lot of phenomenology. So it turns out transgenes are all different. You can get transgenes that are on, you can get them that are off. And for one gene like CDKGFP, you can get, in this case, they're all active every time. Occasionally they're silent, but then they turn, this one seems to inevitably turn on. This one inevitably turns off. So when you first put it in, it can be active, but inevitably this will turn off and all of your strains will be silent. So the order of the foreign sequence inserted in the gene leads to differences in the way they behave. And we still don't know why that is. There seems to be a natural tendency for the GFPs that are on the 5' end to be silenced. And we think we have a little bit of evidence supporting a role for a factor in that process. We just published a paper on that. But anyway, this is just sort of a phenomenon that I think is really interesting. So this transgene, GFP-CDK1, remember that one, that's always off. So what Masaki showed was that you could take a strain that's in this silent state, and you could cross it with an isolated GFP that's in an active state. And what he showed was beautiful dominance. So when you cross them together, the strain that was on goes off. And not only does it go off, but it can be transmitted to offspring in the off state. So we can epigenetically switch the gene from an on state to an off state. It's very, very beautiful epigenetics because there's no difference between this at all, except that now this line has presumably inherited a transitive signal, which is, as we now know, dependent on these small RNA pathways. So this is a funny experiment, actually. I put me up here because when you're a PI, you get to beg and plead and ask your students to do experiments, but they don't always do them, right? So this was one, and I told you we really reluctantly began to believe that the Pee-wee pathway was involved in this. I thought that PRG1 might be upstream of the transgene silencing, but we had already done a genetic test where you take a PRG1 mutant and you cross it with a silent strain, and the silent strain stays silent. It doesn't come back on. It's not required for the maintenance of silencing. And I'm like, oh, don't worry about that. You know, it's when you inject a new one into the strain that it won't go silent in the first place. And nobody believed that that would be true. So, you know, they said they'd do the experiment. They wouldn't do it. Never happened. It was like hard to inject PRG1 because it's almost a sterile strain. It's like really hard. You have to do like 10 times as many injections. So I had to do it myself. And I injected it, and finally I got GFP-CDK1, and it was beautifully on every single time we got an integrated event. I got one, then Heng-Chi got a few, and so we finally, you know, got evidence that PRG1 is required for this silencing phenomenon. So the Pee-wee argonaut plays some role in initiating the transgene silencing. I think probably for me, in my, you know, I don't think I'm smarter than anybody, but I like to, if I can think of an experiment that's, you know, doesn't take too long to do and would have a really fun outcome if it were true, I like to do it. Just, you know, because you have a shot at getting something really interesting. So this was, we couldn't explain why or how PRG1 could do this. It was like illogical in a way. But the idea that this argonaut might be involved as an initiator of the silencing event was something, I couldn't sleep at night until we tested this. So we got this result, and now we're really scratching our heads, how does that work? So the Pee-wee argonaut, we knew from other work, engages small RNAs that are transcribed by the Pol2 polymerase. And this is the weirdest, absolutely weirdest gene family that I have ever seen. There are tens of thousands of piRNA genes clustered on chromosome 4 in the worm. And what they encode, this is showing you an example of a protein coding gene. This is just a typical, you know, snapshot of a genome browser view where you have the protein coding gene and you have these little tiny RNA encoding Pol2 genes that just make a single stranded RNA that's 27 nucleotides long. It's capped, and then somehow it gets processed by decapping, loading onto the Pee-wee argonaut, and then it gets trimmed to 21 nucleotides. So, and the worm has a little promoter element called the ruby motif that's upstream of each of these. So there's 10,000 or more, maybe even 15,000 promoters that are on chromosome 4 that each have a ruby motif. Next to the telomere repeat, it's the second most abundant short repeat in the genome. It's a really weird genomic feature. Anyway, the bottom line is the worm makes a lot of piRNAs. The piRNAs are very sequence diverse. They get loaded onto the Pee-wee argonaut, and the model we proposed was that they scan for targets, and that when they find a target, they can recognize it, you know, in sort of a microRNA-like mode, because GFP has no perfect matches to piRNAs, but if you allow a few mismatches, there's actually hundreds of piRNAs that have a pretty good seed match type targeting of GFP. So then you could, if after that, Pee-wee could recruit RDRP, and then you get this maintenance of silencing. You don't need PRG1 anymore because now you have this memory, and these argonauts are transmitted in the germline. So Pee-wee argonauts are conserved in animals. Humans have them. Humans probably have millions of piRNAs. I don't know what the actual count is. Again, they're transcribed by Pol2 in the germline. They're organized differently in the human, but they're also in mammals. They're really weird genomic features as well. If you take, this is just a detail, but if you take a strain induced in the Pee-wee mutant background and cross it into wild-type Pee-wee, PRG1++, the transgene GFP-CDK1 goes silent, again confirming that the result I told you, that PRG1 is required to initiate the silencing. You take this strain after it's silent, cross it back into PRG1 mutant background, it stays off. So again, confirming that PRG1 is involved in initiating, but not in maintaining the silencing. And there's this notion that it could target with imperfect matches, and so here you've got this part of the scanning mechanism that allows the organism to detect anything that's foreign, potentially. And just examples of, the secondary RNAs are recruited. The RDRP can make lots of additional small RNAs. The piRNA can target, and then you can get induction by RDRP. So we wanted to get further confirmation for this. This is exactly the opposite example. This is Enzi Shen's work in the lab where he did CLASH, and he told me he wanted to do CLASH, and I'm like, no, don't do CLASH, it'll never work, and all you'll get is we think Pee-wee targets every mRNA, so when you clone what CLASH is, you cross-link the argonaut to its target, and you can get the piRNA ligated to a piece of the mRNA, and then you can read out the piRNA targeting for that piRNA by reading out the sequences that are fused to it. And I was the one who was very skeptical it would work. So you do want your lab to not do what you wanted them to do sometimes. I told him, don't do it, it's a waste of time. He did it, it worked incredibly well, it's published. But it's a beautiful methodology because you cross-link in vivo and then you can recover these markers that tell you exactly where the piRNAs are targeting. And some of the statistics here are incredible. He recovered, millions of these CLASH hybrids and was able to, using this method, identify that indeed piRNAs are targeting essentially the entire transcriptome in the germline. So if it's a germline expressed mRNA, it's abundantly targeted by piRNAs. Absolutely all of them are, which again was a little bit surprising because I told you, again, the problem that piRNAs should be, there's too many of them. They should silence everything. So why aren't the self-mRNAs being silenced by this machinery? It's targeting them. Enzi showed that it's targeting them, but they're not getting silenced. But this was possibly explained by this argonaut. Remember I told you that when we read what Caesar1 is targeting by IP cloning of small RNAs, it's targeting all of the animal's own mRNAs. So the notion was that this argonaut could somehow be protecting. And Me Too Seth, a graduate student in the lab who every time she touched an experiment, it worked. And even if it didn't work, it was even more interesting because it didn't work. Me Too's here today. Anyway, so she did this experiment. She was, doing a cross. And, I told you what happens when you cross a silent strain to an active strain. The silent strain dominantly silences the active strain. But when Me Too did this experiment, it didn't work. It was like totally messed up. And she's like, the silent one turned on. And I'm like, are you sure? This was, of course, beautiful, right? This is beautiful. So this we called RNA-A for transactivation. And the question was, well, why did this happen? We still don't know what was special about this transgenic line. We've actually got another paper exploring this, and it's just getting more and more complicated, not less. I won't go into the details of that. But the opportunity this gave her was to test the model using genetics that the Caesar1 Argonaut is required for the transactivation signal. So she was able to do an experiment using very simple genetics. And Caesar1 is a totally essential Argonaut, so if you knock it out, the worms are sterile. Not surprising, perhaps, because it's playing, looks like a very important role. But fortunately for me too, Caesar1 is also haploinsufficient, so just lowering the dose of Caesar1 by 50% completely, I'm not going to go through the details, completely knocked out the transitive activation of a silent transgene. So she was able to confirm with this genetic study that Caesar1 is required for the transitive activation of one silent GFP by a different active GFP transgene. And she also confirmed that the active GFP transgene has Caesar1 small RNAs, RDRP-derived small RNAs, targeting GFP. So in that strain, the GFP sequence has been adopted as a self mRNA. It's now recognized with the Caesar1 pathway, and consequently, it's seemingly protected. And Enzi's data, what really made Enzi's experiment so brilliant, I mean, I couldn't imagine it when he was saying, I want to do Clash, but once we had the data, we realized we could look at we could take all of the pRNA target sites, sort of transcriptome wide, and we could make a metagene analysis of the mRNA region targeted by piRNAs, and we could look at where on the target mRNA the RDRP-dependent WAGO small RNAs are being made. And this was just so striking because right in the middle of the piRNA target site on the endogenous genes, the transposons and so on that are undergoing WAGO-dependent silencing, you see this really robust recruitment of RDRP and this production of these RDRP-dependent small RNAs, and also to some extent at both ends. And Enzi was even able to figure out which of the WAGOs, and different WAGOs seem to bind this. The RDRP-derived peak here is loaded onto one WAGO, and these are seemingly loaded on another to some extent, or at least there's some preferences we just still don't understand. But interestingly, even though PRG1 is targeting the expressed mRNAs, for some other reason we still don't understand, it's not recruiting RDRP very robustly, or at least it's not leading to the production of stable 22G RNAs. There's actually a peak here that is interesting. It may be a Caesar1 associated peak, but there's almost no targeting associated with piRNAs targeting on the Caesar1 or the expressed mRNA. So there's some really interesting biology we don't understand. We don't think it's as simple, as the Argonauts alone. We think there's some other features that allow the organism to know when an mRNA is sort of a good one, that it should be expressed and translated. And those protective mechanisms are somehow being read out by the Argonaut system. But what's really cool about this is that the animal is remembering which genes were expressed. It's remembering all the mRNA open reading frames from not only the open reading frames, but the UTRs are all targeted by these Argonauts. And it's passing this memory from one generation to the next. And admittedly, worms are much higher organisms than humans, but I suspect that maybe, other organisms can do this too. I think what I think is really maybe the take-home message is that RNA can play a very major role in regulating the genome. And I think we have a lot to learn about how the organism is handling information and how it's using RNA to guide information handling, including especially probably in the germline, but I think in lots of other tissues as well. And I'll just show this movie briefly. I don't know if the lights can come down a little bit. I love this movie because it's just showing in real time the mitochondria are in green and these beautiful Argonaut. Now think about it. What I just told you, there's probably almost 1 million different small RNA RISC complexes in these. They're actually like liquid drops in the cytoplasm that are surrounding the nuclei. They're somehow able to look at every transcript. How can they do that? And one of Enzi's results I found really interesting is that even low abundance piRNAs associate with their targets very robustly in an energy dependent manner. So if you have an energetic interaction with a low abundance piRNA, you recover that clash hybrid at a much higher rate even than a much more abundant piRNA that has a lower energy. So it's the energetics of the scanning here is fascinating, and the fact that the organism can actually use its Argonauts this way to look at every single transcript, probably as it's exiting the nucleus, which is why you probably want to put them right there at the nuclear periphery, and they're actually some evidence they're associated with nuclear pores. One of the things you can also see in this movie is the yellow, where the mitochondria and it's really cool. I have no idea if there are Argonauts that are scanning mitochondrial gene expression and maybe genome maintenance or something could be going on. But there's some really fascinating biology going on here because this is weird stuff. It's really cool. I think there's a bright future for RNA biology. And I haven't said anything about therapeutics yet, but it's very near and dear to my heart because my daughter, Victoria, is type 1 diabetic. She's totally dependent on insulin to be alive. And in this picture at the Nobel headquarters in Stockholm, she's gathering up these great gold medals, and they're full of chocolate, of course. To eat them, she needs to give herself insulin, and she needs it every single day of her life. I think the future of oligonucleotide therapeutics is like the future of people like my daughter, who would literally not be alive if not for the brilliant work of people like the people in this room who are trying to make oligonucleotide insulin for people who are going to be, extremely grateful for the opportunity to have a life. I mean, she can have she's 18 now, and she's having a normal, happy life. And sure, it's onerous to have to deliver insulin every day. It's not a cure. But this is the kind of opportunity I think we have to transform the lives of patients and give them a future. And I really thank all of you for all the great work that you've done and are doing to further that goal of really making a difference in people's lives. Because whether you realize it or not, that's what you're doing by studying RNA biology. So thanks. I hope I didn't go over too much. Am I good? Whew, that's like the first time I've ever...
Thank you, Dr. Mello, for a great talk. We have time for one quick question.
Oh, okay. I didn't do that good.
Yeah.
Go, Victor.
Right, you mentioned that CSER is dosage sensitive. Is PRG also? Can you see that they actually are balancing them?
Yeah, that's a great question, and there is evidence that they can balance each other. This PRG1 mutant is not haploinsufficient, but we do know that if you increase the piRNA targeting on a gene, you can sort of force it to become, more silent. So we can play around with that, and we can definitely. There's a lot of evidence that there is this kind of a competition going on.
I mean, it feels like each RNA is experiencing a kind of switch.
Yeah, that's one of the things I think is really interesting. I mean, I think that, at least in the germline, RNAs can experiment with whether they want to be silenced or not, right? By changing the sequence of the RNA such that it's targeted more or less by piRNAs, an mRNA could essentially become epigenetically switched on or off. And we have evidence that these pathways are involved in regulating genes. They're regulating normal genes that function in normal biology. And one of the ideas that I think is really attractive from an evolutionary perspective is that organisms could sort of experiment with turning genes off using an epigenetic mechanism. And if there's some advantage to that, of course, there's no shortage of nucleotide mutations that could perhaps permanently enforce the decision, now that gene's going to be off, or maybe just tweaking gene expression with these mechanisms. That's probably why some of these genes are essential, because they're actually regulating many endogenous genes. So I'll stop there, and thanks again, and I hope everyone has a great meeting and look forward to chatting more with everybody.
Thank you, Dr. Mello.